Okay, so in exercise C, we're going to observe the action of an enzyme. We've been talking about enzymes and enzyme-driven reactions. In this case, what we're going to do is see if we can observe the enzyme called amylase that will work to break down a starch molecule from that polysaccharide level to the simple monomers that it's composed of. And so what we have here is a solution that has amylase enzyme in it, and we have a solution of some starch molecules. We're going to mix these together and have them ready to go in the proportions as directed in the lab manual. And then we're going to set up a series of test tubes that we're going to utilize to observe the action of this enzyme over a period of time. In each of these test tubes, we're going to have water and an indicator that we've learned about, Lugol's iodine, that again will give us some indication of the presence of starch in the test tube when we get the experiment rolling, okay? And we're going to have test tubes set up that we're going to use at different increments of time with the iodine and the water ready to go, that we will be adding the starch and amylase solution to at the different time increments. When we get the starch and amylase solution ready to go, we're going to be timing how long we've allowed the amylase to potentially do its thing, and at each time increment, we're going to take a sample from this starch and amylase solution and add it to each one of these test tubes that represent the different time steps that we want to check and see if that enzyme activity has happened and to what degree it's happened. Okay, so we've gone through the time steps, adding the starch and amylase solution to each of the test tubes that we set up ahead of time at the different time increments. And as you can see, we've got a little bit lighter color of that reaction of starch and iodine indicator from the very earliest time step toward the latest time step as the starch has had more and more time to do its thing in that beaker that we set up at the beginning. And of course, we have controls at either end to compare these results to. Now, you can visually see that there's less and less reaction of the indicator with iodine and starch as we get longer and longer time period that the enzymes had to work. But it's also nice to have a little bit more precise results of how much of that activity has occurred. So to do that, we're going to use our spectrophotometer again. And what we'll do is take a sample in the small cuvette that will fit into the device of each solution of each time step that we've got now prepared, and measure how much absorbance at the proper wavelength that I believe we're going to use 540 again, getting through that solution. And as we've learned before, the darker the solution is, the more that light being passed through is going to be blocked or absorbed from getting through. And we can get precise measurements of how dark or light that indicator has become with what starch was left in each tube in a more precise way than just kind of guessing, well, it's a little lighter color or a little darker color in a precise way.